I am at the lab of forest genetics in the Thünen Institute. Today I will learn how genetic timber tracking and species identification work in practice. This is Susanne Bein. She's part of the technical staff here. After cleaning everything, Susanne scrapes the upper layer of a wood sample away. She needs to get intact DNA. These scraped chips of wood from the sample have to be filled into a little plastic tube. This tube already contains tiny ceramic petals. These little ceramic petals function as a mortar because they are crushing the wood chips. At the end, the chips of wood should be pulverized. Centrifugal force and a very fast spinning machine will support this process. To be able to walk around with the samples, the little tube needs to be put into a sterile plastic box. Susanne says that it is super important to guarantee sterility because genetic wood analysis is comparable to forensics. She has to make sure that the sample is not contaminated. People working in a lab have to be very strict about this. Now we're going to the lab next door to pulverize the sample. Therefore, Susanne needs to get liquid nitrogen. This machine is called a Bead Ruptor. Bead Ruptor because it's supposed to pulverize the little wood chips. The plastic tube will be spinning around here for 1 minute. To support this process, Susanne cools down the chips with liquid nitrogen. The Bead Ruptor is a great solution for releasing DNA from very tough samples, while still retaining molecular integrity. To support the extraction of DNA in the following incubation time, Susanne adds a buffer to the wood sample. Now the buffer has to be put into the little plastic tube. Wood DNA analysis is a relatively new method that is still improving and expanding its reference database. Developing reference data is a huge work, where the DNA of a species has to be analyzed in the search for genetic markers that are distinct between species or geographical locations. But now back to the analysis. The wood sample and the added buffer are mixed now to prepare the extraction of DNA. Finally, the sample needs to be warmed up in a heating block for 5 hours. The right temperature is 55 degrees. During these 5 hours, the buffer has enough time to decompose all kinds of cell walls and the cell nucleus to be able to extract the wood DNA. Susanne puts the sample into the heating block, where it spends 5 hours shaking around. The lab expert says they are hoping to develop DNA markers with this method. With the help of these markers, they aim at specifying not only the wood species, but also the origin of the timber. Ideally, they can then tell whether the wood comes from Africa, South America or Asia, and sometimes even the region, to guarantee a reliable and precise classification.